Today's presentation is over the West Nile virus. Several characteristics of its morphology is an isohedral enveloped virus of 40 to 50 nanometers in diameter. It is also a single-stranded positive sense RNA genome. The gram stain for a West Nile virus is that it has a negative result. Next, we will cover the history of the West Nile virus. The West Nile virus was first isolated from a febrile patient from the West Nile district of northern Uganda in 1937. The patient presented in the setting of a large epidemic study of yellow fever virus. However, inoculations of mice with the patient's serum resulted in the isolation of a virus with physical and pathological properties similar to those of the two flaviviruses, St. Louis encephalitis virus and Japanese bee encephalitis virus, and sharing immunological relationships with these viruses. Although the index patient presented with fever only, these first studies were the newly discovered virus indicated that pathology primarily involved the central nervous system. The epidemiology and ecology of the West Nile virus was first characterized in detail during several outbreaks in the Mediterranean basin in the early 1950s and 1960s. The first recognized epidemic of West Nile virus occurred in Israel in 1951 in a small town outside of Haifa, where a total of 123 cases with no fatalities occurred among 303 inhabitants. Young children represented the majority of the cases. During this outbreak, the various clinical features associated with the infection were first described in detail, with the main symptoms being fever, headache, myalgias, anorexia, abdominal pain, and vomiting, and diarrhea were somewhat less common. Several large outbreaks in Egypt between 1951 and 1954 led to a further understanding of the ecology, epidemiology, and clinical characteristics of the West Nile virus. By the time the virus was first detected in North America in 1999, it had already had a recent history of more frequent outbreaks and more severe illness. In late August of 1999, a cluster of severe cases of encephalitis was noted in an area around Queens in New York City. An epidemiological investigation by the New York City Department of Health identified 8 such cases and revealed that all the patients had previously been healthy and had resided within the same 16 square mile area and had recently engaged in outdoor activities. During the summer of 2002, however, the numbers of West Nile virus cases in North America was unprecedented. This was the largest outbreak of the West Nile virus ever recorded. The West Nile virus expanded its geographical range from the Mississippi River area at the conclusion of 2001 to the Pacific coast by the end of 2002. As of January 2003, the provisional human case count for the 2002 season was 4,156, including 284 deaths. Particular regions of the U.S., including parts of Louisiana, Mississippi, and the Chicago area, saw a particularly high number of cases. Severe CNS disease continued to be predominantly seen in older individuals, but more cases of milder febrile illness in younger patients were detected, possibly as a result of enhanced surveillance efforts. The ecological role for the West Nile virus is very difficult to explain, and to even see if there is an ecological role other than the destruction of cells that it affects. It harms the mosquitoes as well. One possible ecological role that can be viewed as a positive is that through the people that unfortunately get infected by this disease, there might be a possibility of using their experience to develop some type of preventative treatment, such as a vaccination. The metabolism of the West Nile virus is centered around acetyl coenzyme A, as a central metabolite for both the biosynthesis and catabolism of fatty acids. Although the steps in the biosynthetic and catabolic schemes are closely related, there are specific differences. The individual steps are also independent by their spatial separation in biological cells, as biosynthesis occurs in the cytosol, while catabolism takes place in the mitochondria. The common fatty acids result from the sequential 2-carbon elongation of acetyl-CoA by a series of enzymatic reactions utilizing one or more molecules of acetyl-CoA. The two-step elongation scheme of fatty acid biosynthesis is catalyzed by either discrete enzymes catalyzing specific steps or by a large multi-enzyme complex able to catalyze all the steps in the pathway. The life cycle of the West Nile virus is quite interesting. The entry of the West Nile virus is through receptor-mediated endocytosis after virus attachment to the cell surface. Within the late endosome, the envelope protein will undergo a conformational change, resulting in fusion of the viral lipid membrane with the endocytotic membrane and the release of viral RNA genome into the cell cytoplasm. Following capsid dissociation, the RNA genome is replicated and the virus assembly is initiated following a well-documented program. The viral polyprotein is translated and processed on intracellular membranes, resulting in the expression of the 10 viral proteins. The original viral RNA is replicated by viral and cellular proteins onto multiple copies to be used in the production of new virons. The structural proteins assemble onto membranes in the endoplasmic reticulum and associate with the nucleocapsid and bud into the cytoplasm via the Golgi network. The virus travels to the cell surface in an exocytotic vesicle and matures as cellular enzymes cleave the PRM, resulting in the release of a mature virus from the cell surface. The West Nile virus has a very interesting molecular biology. There are several special proteins involved in its survival. The TLR3 protein, better known as the toll-like receptor 3 or CD283, plays a fundamental role in pathogen recognition. It activates an innate immune response. They also recognize pathogen-associated molecular patterns that are expressed on infectious agents. It also controls cytokines for effective immunity. The protein forms in a large horseshoe shape with sugar molecules covering most of the protein, and it is highly positively charged. The WNVE protein specifically blocks the production of antiviral and pro-inflammatory cytokines induced by dsRNA in macrophages. The RIP1 protein is responsible for cell death. When the protein binds its ligands, they aggregate and start a signaling pathway that either triggers an inflammatory response or, unfortunately, cell death. The PRM protein is important in the formation and maturation of viral particles. It consists of 7 antiparallel beta strands stabilized by 3 disulfide bonds. The protein acts like a cap protecting the virus until its replication is completed and released from the cell. It also protects the hydrophobic fusion loop of the E protein. Now, since all the molecular biology has been discussed about the West Nile virus, there are several important general announcements that should be known about the West Nile virus, especially how it is transmitted. The first step is transmitting the West Nile virus occurs when the mosquito bites an infected bird or animal, from which the mosquito feeds on its blood where it obtains the virus. It is transmitted again each time the mosquito feeds, which means if the infected mosquito bites you, it will cause you to become infected with the virus. Some of the basic symptoms of the West Nile virus include fever, headache, body aches, skin rash, and swollen lymph nodes. However, there are more serious symptoms, such as stiff neck, disorientation, convulsions, coma, paralysis. However, in most instances of the West Nile virus, they are mild and go without needing intervention from a medical doctor. Currently, there is no treatment for the West Nile virus, or is there a vaccine available. In the more severe cases of the West Nile virus, the patient is hospitalized to obtain intravenous fluids and high-powered pain medications via IV. Several ways to try to prevent the spread of the West Nile virus is to avoid mosquito bites, and to also use insect repellents when you go outdoors and into the woods. And if the weather is not too hot, wear long sleeves and pants. Another prime point is to avoid the prime biting hours for mosquitoes, which occurs primarily at dusk and at dawn.
Kill the mosquito.